Once a B cell recognizes an antigen, it becomes activated. It expands clonally and starts producing antibodies specifically directed against that antigen. Antibodies are composed by two distinct functional subunits: the fragment of antigen binding, which confers specificity to the antibody, and the constant fragment, which has effector functions. Monoclonal antibodies are produced in vitro in order to recognize specific antigens and to induce distinct effects on target cells. A class of monoclonal antibodies has a direct cytotoxic effect on cancer cells. For instance, some antibodies can bind specific tumor antigens that activate molecular pathways able to induce apoptosis. Other antibodies recognize tumor antigens that block the cell cycle and proliferation of cancer cells. Moreover, some monoclonal antibodies can be conjugated to toxins. After the binding between the antibody and the antigen, the toxin is released and acts locally, killing that tumor cell. Another class of monoclonal antibodies carries out an indirect cytotoxic effect on cancer cells. This function can be immunomediated, in other words, mediated by the immune system. For example, the interaction between the antibody and the antigen can activate the complement components, which in turn release cytotoxic granules inducing lysis of the cell. The binding between monoclonal antibodies and tumor antigens can induce the recruitment of macrophages and natural killer cells, causing the same result. In other cases, the interaction between tumor antigen and monoclonal antibodies unmasks cancer cells from T cells. Another indirect activity of monoclonal antibodies targets the tumor microenvironment and blocks molecules involved in angiogenesis, thus reducing nutrients for cancer cells. The main limitation of using some monoclonal antibodies relies on drug resistance, which is the ability of cancer cells to become resistant to a specific drug. Monoclonal antibodies are useful immune therapeutic agents for the recognition and elimination of cancer cells.